and we need some alternative of that, and I will talk about that what can be the alternative and some of the backgrounds of my research work. So I'm greatly grateful to my supervisor, who is Rita Roy, and my whole team, who is from UK originally, and now I'm going to my talk. So firstly, we all know that we are using plastic and in every day. In agriculture, we use it in mulch and in other materials, in medical instruments, including catheter, syringe, and many other instruments, food packaging, and many more industrial uses, including water bottle, which we are regularly using, and the water pipe. So what are the consequences of this use? It actually causes many detrimental effects. First of all, land pollution. So we all are aware about this land pollution, which is very toxic, and when it is going for burning, it becomes it gives us the air pollution, and then also it causes marine pollution. So all over it is detrimental to our health, to our environment. So we need to think some of the alternatives of the plastics. So besides this, air pollution and other pollutions, it's also being carcinogenic. How? Because the bacteria also grow inside that, and also all the pathologists and pathogenicists are aware about that. Bacteria grows inside that and cause biofilm, and which can be carcinogenic, and also it causes the post-operative infections, and which is very detrimental for our health, and even it can cause the death. It has been found that due to the antibacterial resistant bacterial pathogens, many people are dying, and in a report it has been found that E. coli, Staphylococcus aureus, and others actually cause millions of deaths. So it is a high time to think that how we can actually restrict this. So as an alternative, there are some biodegradable polymers which is called polyhydroxyalkanoates, which is a synthesized polymer naturally by the bacteria, and it is more importantly biodegradable, non-toxic, and it has multiple components, means according to the bacterial strength and the source of food, means what is the carbon source, it actually can be differed in a various constituents, and therefore it has a wide range of mechanical properties which can give us a way to get rid of this plastic and go for a biodegradable materials to use in our environmental friendly to replace the plastic. So among these polyhydroxyalkanoates, in short PHA, there are two types. One is mainly long chain and short chain, and among the short chain, poly 3-hydroxybutyrate, which is shortly P3HB, is widely used because it is rigid, it has good oxygen permeability, and it is UV resistance, as well as when it is breakdown, actually our body can produce it. It means from the liver, it produces the 3-hydroxybutyrate. So it is completely risk-free for the health. So our research objectives were to develop antibacterial polyhydroxyalkanoates, and then to investigate its thermal, mechanical, and surface properties, as well as to investigate its biocompatibility and biodegradability, so that we can get an insight that how it can work against the commonly used plastic. So as a material, I used Bacillus subtilis OK2 to produce this P3HB, and as an antibacterial material, I used Eucalyptus globulus essential oil, which is commercially bought from the Biorama. And bacterial, I tested these polyhydroxyalkanoates incorporating this essential oil against Staphylococcus aureus and E. coli. And further, I tested the biocompatibility against the mouse muscle myoblast cell line. Sorry for this it is called incompatibility. So in a method, I have used batch fermentation to produce the P3HB using the chloroform extraction method, and further I tested that if it is correctly P3HB or not, FTIR and GCMS, And further, I have also done the characterization, means thermal and its physical properties using the DSC and tensile testing. And the antibacterial assay has been done using the agar distribution assay as well as the minimum inhibitory concentration. So further, after incorporating the essential oil into the P3HB, I have again run the same analysis, means I have seen that what are the antibacterial properties, what are their mechanical and physical properties, and further I have done the in vitro analysis for the biocompatibility assessment. So here is the fermentation profile. So from the profile I can see that glucose consumption has been reduced over the time, means 48 hours time, and also the OD is increasing, which means the bacteria is growing and it's about to die, because if you look at the DOT, it's going to down, means the dissolved oxygen is also going to down. And further, after producing the P3HB, I have checked its properties actually matched with its component at 170 mZ, which has been done in GCMS, and further by the NMR also I have confirmed that it is P3HB. And then I have tested the antibacterial properties of the eucalyptus oil, and I have found that eucalyptus oil show in 2 centimeter halo against Staphylococcus aureus and 1.5 centimeter halo against E. coli, while the positive control was higher, because we assume that because it was in agar-test method, maybe there are some problem in diffusibility which needs to be optimized in further. And further, we have done some minimum inhibitory concentration to see that what are the minimum inhibitory concentration of the bacteria using this essential oil, and we found it that in Staphylococcus aureus it was 125, and for E. coli it's also like 125. So we used this MIC value for further analysis. And further, we incorporate this eucalyptus oil into this P3HB is a solvent casting method, and I actually tried in different ways, in open lid, closed lid, and like semi-closed lid, and I found good result with the closed lid method. And again, after incorporating this essential oil, I tested if it is incorporating and if it is actually P3HB and E3 is presenting there. So I found that yes, the P3HB properties was CO, and it is the P3HB plus CO, means it is also present here, and the other properties of the eucalyptus oil which has been separately done, so all are present here. That means that eucalyptus oil incorporated into that polymer successfully. So here is the photograph of that agar-disc method. After this is the net P3HB. This is the P3HB after incorporating the eucalyptus oil. So we can see the clear differentiation that the net P3HB does not show any kind of halo, means any antibacterial activities against the bacteria, while P3HB with the eucalyptus oil showed clearly antibacterial properties against those both of the bacteria. Again, after checking this the thermal as well as the physical properties of those polymers, it is very essential, because if we want to use those polymers for our regular use for agricultural mulch or in medical devices, it needs to improve its properties, because only having the antibacterial properties is not the criteria to replace the commercial plastics. So we found, interestingly, that the thermal properties of P3HB and EO actually improved. So the melting temperature increased, crystallinity decreased, which means essential oil has some electrostatic or like elasticity increasing properties, which actually favors this P3HB because it is crystalline, and due to being crystalline, net P3HB cannot be used in tissue culture or any kind of scaffold producing. So being soft, it can be used in other like soft tissue engineering and scaffold making. And also the mechanical properties improved, means its tensile strength reduced, elongation at the break increased, and yielding also increased, means its stability increased. So, incorporating the eucalyptus oil not only improved its antibacterial properties, it also improved its mechanical as well as thermal properties, which is very important for any kind of polymer to use in a diverse way. So, further, I have also seen its surface quality using the ACM. I found that the roughness of P3HB after incorporating the eucalyptus oil increased, which actually improved its protein absorption, which I will show in a short time. And also its water angle reduced, means its hydrophobicity also reduced, means its hydrophilicity increased, which actually favored the biodegradability. So in vitro compatibility study, we found that the cell proliferation and cell viability actually not very much promising, which decreased, and that could be the future study that how we can improve or by optimizing the oil concentration so that the biocompatibility can be increased. And in case of again in protein absorption that I have already mentioned, that due to the increase of roughness of the surface of the new P3HB, it can absorb more protein, which is also very important. So if we want to make any scaffold, it needs to be improved the protein absorption quality. And again in the biodegradability, checking the biodegradability properties.
So pretty interruption, your time is already over, so you have to complete within 1 minute.
In case of biodegradability assessment, we found that the biodegradability actually improved in case of new polymer. You can see here that though the pH is not very much different, but the water absorption quality improved and also the weight loss improved, means it can be degradable more quickly than the net P3HB. So, in a conclusion, it is a new polymer which has the multiple qualities, including the antimicrobial quality and improved mechanical as well as thermal quality, which can give it a way to use it in a multiple ways in agriculture, medical as well as food packaging. So, after optimizing its concentration into the incorporating into the polymer and assessing the cytocompatibility, it can be widely used. Thank you so much.